So good morning, everyone. Today it's my pleasure to introduce Dr. Baroukhim. Dr. Baroukhim is a physician scientist at the Harvard Medical School and the Farber Cancer Center. It's an institute. Helped us understand the genomic alterations that are under positive or negative selection. And he used these insights to improve patient care. For example, his group has developed a tool called GISTIC, which is a fundamental tool or foundational tool that identify recurrent copy number changes in cancer genome. And more recently, his group developed another tool called BISCUIT, published in Nature, which pushes our ability to analyze cancer genomes even further. So please join me in welcoming Dr. Baroukhim.
Great. Thank you so much for that kind introduction. First off, I should say that I'm very honored to be giving this talk. Often folks wait to the end to ask questions for the talks like these, but I encourage you, if any questions come up, please interrupt me and ask them. It can be the case that talks on Zoom can be a little sort of distancing for folks because you sort of just see the speaker and nobody else. So for those of you who are junior, what I tell the people in my lab is that if you turn your camera on, then everybody in the conference or the meeting gets to see you, and you become more recognized, right? So I always recommend that at least the junior folks and maybe everybody turn cameras on. So, and from the speaker side, that's helpful as well. And then do ask questions that will also sort of get you recognized. Anyway, so I'm going to talk today about detecting positive and negative selection and shaping rearrangements in cancer genomes. Before I start, let me just see here. I just want to say that I do, in terms of conflicts, I don't have any real conflicts, but these are some companies that I'm associated with. In general, our laboratory works on a variety of things. I see patients with brain cancers, and so we do a lot of work on brain cancers. We also do work on tumor evolution, and then a lot of work on alterations in chromosome structure, which is what I'm going to focus on today. And when you think about the cancer genome, including alterations in chromosome structure, the first thing that strikes you is that the cancer genome is complex, right? So this is an example of a single cancer genome where the lines in the middle indicate rearrangements in different loci. Around that, there's copy number alterations in this sort of outer sort of middle rim or whatever. I don't think point mutations are even indicated, but this particular cancer has thousands of point mutations. So there are many events, right? And we know that only to transform cells into cancer, you only need maybe 5 or 6 genetic alterations. So why do you have so many events? And for many of you, this is sort of a review, but most of these events are likely to be passenger events, right? So as cancer cells divide, cells divide, they gain mutations at random throughout their genome. Most of those are selectively neutral or deleterious, indicated by the yellow and blue here. But in some cases, a set of mutations can occur, the drivers that transform the tumor, the cell into a cancer, and then the cancer takes off. And those transforming mutations occur in the context of preceding passenger events, which could be selectively neutral or deleterious. In this case, I indicated a deleterious mutation because, in fact, those can be fixed in the genome. So although that might not be expected, right? And then as the tumor cells continue to divide, you get more mutations, including drivers and passengers, shaping further selection. So, much of cancer genomics has been dedicated to detecting the driver events. And as Ruping Sun just mentioned, we developed GISTIC a couple decades ago now to try, which was one of the first methods to distinguish driver events from passenger events, right? So the rationale is that passenger events can occur anywhere in the genome, whereas there's only a limited number of events that can drive cancer, right? So that you tend to see the driver events recurrently, and the passenger events would be observed at much lower rates. So the approach to GISTIC is that you take individual cancer genomes and identify where, in this case, copy number alterations lie, and then you sort of add up the copy number alterations at each locus to generate a summary score across all the cancers, right? And then you go back and you sort of model, well, or simulate what would this have looked like if these were all passenger events. So assuming that these were all passengers and could have occurred anywhere in the genome with equal likelihood, what kinds of scores could we have gotten? And so you use that to generate a distribution of scores, and you apply from that distribution, you determine a significance threshold beyond which you wouldn't have expected scores to be achieved by chance. And then you apply that to the observed data to identify loci that were altered more often than you'd expect by chance. So this has been used to identify a bunch of driver events in copy number alterations in cancer, and similar methods have been developed for point mutations. So, for instance, this is how MutSig works. It looks at point mutations at various loci in the genome, and again, as the number of point mutations per locus to identify loci that are mutated more often than you'd expect by chance. And these methods have been very successful. So, for example, as of 12 years ago, we had 224 cancer genes that were detected across 21 tumor types by MutSig. And so we've had a really great, I think, process of discovering cancer genes, especially from mutation analyses of point mutations in small indels, where at this moment, most people think that we've identified almost all of the prevalent driver point mutations, at least in the common cancers, and so the work goes on from there. But if we think about what's important in cancer, well, point mutations occur in about 1 in a million bases in the average tumor, right? So at 0.0001% frequency. Whereas copy number alterations alter about 1/3 of the cancer genome. About 20% of that is arm level alterations, which affect whole chromosome arms or whole chromosomes. And then another 12 or so percent is due to focal events within chromosome arms. And so here we have several orders of magnitude difference between copy number alterations and point mutations. So when I look at this, my conclusion is that for those of you who know people who have worked on understanding point mutations in cancer, their lives have been wasted. Clearly the most important work is going on in copy number, and I say that as a copy number person. So how well do we understand copy number alterations? Well, we developed GISTIC. We've run it across many thousands of cancers. This was an older paper which identified 140 regions of amplification or deletion with many of the most important oncogenes and tumor suppressor genes in cancer identified in those regions. But I would argue that actually we understand very little about what's going on in copy number changes in cancer. And the reason for that is that the most common events are often not understood. And so this is a listing of the most common genetic alterations in cancer. Well, so the most common one is mutations of P53, right? So P53 is the most important gene in cancer. But then the 24 next most common alterations in cancer are all copy number changes. And remember that copy number changes affect a third of the cancer genome. So whereas point mutations affect 1 in a million bases. So it stands to reason that the most common genetic alterations will be copy number changes. And among these 24 common copy number alterations, 23 of them affect whole chromosomes and whole chromosome arms, right? So they're amplifications or deletions of whole chromosome arms. Those we really don't understand well at all. Each of these events can affect hundreds to thousands of genes, right? And so in some cases, we know that there are driver genes on those chromosomes. So for instance, 8q amplifications are very frequent. They cover MYC, which is one of the most important amplified drivers in cancer. But even in the case of 8q amplifications, well, we know MYC is driving, but then another approximately 1,000 genes are being altered. What are those? Are any of those other genes providing any selective benefit or disadvantages to the cells in which they occur? We don't really understand that. And all of these starred chromosome arms are ones where we really had a hard time identifying a gene that would explain the frequency with which those alterations occur, right? I mean, some of these cover known cancer genes, like 7p covers EGFR, but EGFR is focally amplified in very few cases where you have the 7p amplifications. Other regions like the 17p deletions, for instance, encompass P53, but that one's wrong. Well, some of the deletions encompass known tumor suppressor genes, but those tumor suppressor genes are mutated in very few of the cases where those deletions occur. So, we really don't understand anything at all. I would argue almost nothing about the starred events, and even the unstarred ones, we understand very little. So why has it been so hard for us to understand the impacts of copy number alterations and what's causing those impacts? Well,
hey, Rameen,
yeah.
this is Andy Nelson. I hope you don't mind, but you did invite questions throughout, so I wanted to just ask a quick question on this. And I can't remember. I was trying to look up your paper that you're showing here right now. But when you guys define these copy numbers across this entire group of cancers, can you kind of describe what the range of quote unquote amplification is? Again, as molecular pathologists, we obviously think a lot about the difference between 1 or 2 or 3 copy gain versus, like you kind of alluded to with EGFR, focal amplifications, particularly extra chromosomal amplifications that start to get to copy numbers that are in the 20s, 50s, hundreds, just
right.
want to get that sense from you because I can't remember.
That's a great point. And it's been, I think, a bit of a controversy in pathology over decades as to whether polysomy, the gain of 1 copy or 2 copies is really a drive, could be a driver event. It could really affect the tumor evolution, or a hemizygous loss alone. How much is that affecting tumor evolution? Whereas high-level amplifications and homozygous deletions, have been widely recognized as being, driver events. In general, alterations of whole chromosome arms do not achieve very high levels of amplification or deletion. You cannot delete both copies of an entire chromosome arm and have the cell survive. Every chromosome arm has essential genes. Likewise, it seems that if you, subject a whole chromosome arm to gains beyond like 4 or 5 copies, it becomes toxic. And so these events tend to be lower level, the whole arm events. The P16 deletions, the 25th most common thing, are homozygous deletions. And then we have, the most common high-level amplification is cyclin D1 amplification here, that's listed as well. So this talk will describe our attempts to understand both the low level and the high level, but you're right that there is a distinction. Did that address your question?
Absolutely. Thank you. Appreciate it.
Yeah, no worries. Again, I'd love to encourage anybody else to ask questions because that makes me recognize that people are listening. So so why don't we understand these events? Well, one thing is that they cover so many genes, and so often it's hard to disentangle, which gene is doing what. But along with that is the issue that the events themselves entangle the effects of different genes, and that can affect their frequencies. And so let me just show you an example of what I'm talking about. Here is a view of a part of chromosome 9 across many, shown along the vertical axis, among many cancers shown along the horizontal axis. And regions in blue are deleted, and the darker the blue, the deeper the deletion. So these dark blue areas are homozygous deletions. This is the region that's the region surrounding P16, which in the prior slide, was indicated as the most frequently deleted region in cancer. And so we know that P16 and its neighbor P15 are important tumor suppressor genes because this is so frequently deleted, right? But if you look at these deletions, they can extend quite far from P16, all the way out to this location here, which just borders this gene SMU1. SMU1, it turns out, is a gene that regulates RNA processing and is cell essential. If you knock out SMU1, you kill any cell, right? So it makes sense that none of these deletions actually encompass SMU1, because if they did, they would kill the cell, right? So if you think about it, the reason that P16 is deleted as frequently as it is, the reason that we think P16 is so important is because SMU1 is as far away from it as it is. If SMU1 were closer to P16, cells would have fewer options to delete P16 and maintain viability without deleting SMU1. So you would see fewer deletions of P16 and fewer cancers. And so you'd think that P16 was less important, right? And so to understand the effects of positive selection, you really need to understand the effects of negative selection and how that's constraining the cell's ability to take advantage of positive selection. So in contrast, here's NF1, which is also a potent tumor suppressor gene. I remember studying it 20 years ago when we were developing the GISTIC method, and GISTIC did not identify it as a locus that was that underwent recurrent deletions because the deletions were too infrequent. I remember a colleague came up to me, Karen Chakowski, and said, well, we've identified, we have reason to think that NF1 is an important tumor suppressor gene in sporadic glioblastoma. Do you have any evidence that it's deleted? And I said, well, we see occasional deletions, but they're not significant, statistically significant. And she said, oh, great, you see deletions. So we'll call it, we'll use that to support our hypothesis. And I said, sure, but I don't really believe that because we're not seeing them as statistically significant. Well, she was right. I was wrong. NF1 is an important tumor suppressor gene in glioblastoma. And the reason we don't see frequent homozygous deletions is because it's surrounded so closely by these two essential genes, UTP6 to the south and PSMD11 to the north. So a deubiquinase and a proteasome component. So cancers really have very few options to delete NF1 while not killing themselves, which is explains why it's so infrequently deleted. And when you see NF1 inactivation, it's more frequently due to point mutations in the context of loss of heterozygosity. If you look further, well, this was I mentioned the study of 5,000 cancer genes, with GISTIC that we did some time ago. We've extended that to 11,000 cancers. This is the list of the most frequently deleted loci, right? P16 comes up at the top, and we have a lot of fragile sites. RB1 it comes out at number 7. There's a lot, another locus that we don't understand. And then P53 comes out as the 35th most significantly deleted gene in cancer. P53 is the most important gene in cancer. So why is it only the 35th most frequently or significantly deleted gene? One of the reasons is because of its surroundings, right? Immediately to the telomeric end of P53 is RNA pol IIa, right? 180 kilobases away. And if you look at the dependency map, CRISPR screening data against, well, in this case, it's actually just RNAi data against RNA pol, RNA pol II. Like knocking it down is extremely lethal to cells, all cells. And the same is true to the right, where you have aurora kinase B half a megabase away, where, again, if you knock it out in CRISPR's data, all 1,000 cells sort of die immediately. All 1,000 cell lines that have been treated in this way die immediately. So it's surrounded by these heavily essential genes that are just right up against it. And that makes one wonder whether this is a way that nature is using to prevent cancers by making it harder to delete P53. But the result is that we don't recognize, from copy number analysis alone, the importance of P53, in part because of the negative selection going around it. So that, to me, suggests that negative selection needs to be understood. Now, folks have already tried to understand negative selection in cancer genomes, and that includes Inigo Martin-Carena, who at the time was in Peter Campbell's lab, who published a paper in Cell, and Shamil Sunayev's group, who published similar results in Nature Genetics that same year, using different methods to identify negative selection. And so Martin-Carena used an approach of looking at the ratio between non-synonymous and synonymous mutations in a gene. And the thought was that, if you mutate a gene randomly, on average, there should be about 3.5 non-synonymous mutations for every synonymous mutation. And so if you see a ratio of 3.5 to 1, that's consistent with random mutations occurring. But if there are more non-synonymous mutations than you expect relative to synonymous, if this ratio skews positive, above 1, then then that indicates that cancers are trying to select for the non-synonymous mutations. So that's a driver gene. There's positive selection going on. Whereas if you if you see fewer non-synonymous mutations relative to the number of synonymous mutations, that indicates that purifying selection is going on, eliminating those some of the non-synonymous mutations, right? And the reason, and so that's negative selection. And so the result is that across all cancer types, you tend to see positive selection in point mutation space and not really much negative selection. And so there are something like almost 3% of genes that appear to be undergoing positive selection by this kind of these kinds of criteria, whereas less, about 2%, 0.2% of genes might be undergoing some level of negative selection according to these criteria. So very little evidence of negative selection. Why is that? One possibility is that point mutations only inactivate one copy of the gene, and so you often have the other copy. And another possibility is that we just don't have the power with point mutations because they affect so few genes in a given cancer. But copy number alterations seem to be different. We see substantial negative selection there. And so that leads us to the recognition that there's both a problem and an opportunity in the context of negative selection and copy number alterations. And the problem is that you can't understand positive selection without accounting for negative selection because of the entanglement. But then the opportunity is that you can use evidence of negative selection, including through entanglement situations, to identify genetic events that cancers don't like, right? And so you could imagine that therapeutically that can be very interesting if you know that there's a gene that a cancer does not want to lose. That immediately makes one sort of think about, could it be a leverage for therapeutic opportunities? So we're spending a lot of time trying to think about how you can understand negative selection in cancer genomes, or detect effects of negative selection in cancer genomes in copy number space. And looking back at this example of the p16 deletions, the way we sort of noticed SMU1 was that it was beyond the boundary of all the homozygous deletions, right? So for negative selection against deletions, you want to look for genes that are outside the boundaries of the deletions that do occur, right? And similarly for toxic genes, you want to look for genes that are not amplified, that where there, when there are amplifications, they stop before the gene. And so we really need to do boundaries analyses rather than just look at which loci are most frequently deleted. So that leads one to sort of ask about, well, how, what determines the boundaries of copy number alterations? Well, this is a, view of a region of a cancer genome that had undergone whole genome sequencing. And the vertical axis shows the sequencing coverage at each of the loci along the genome, and the horizontal axis is the genome. And the sequencing coverage tends to correspond to the number of copies of DNA at the locus, at each of these loci. And so as you go along the chromosome, you see that there's a drop from 2 copies to 1 copy, and then it comes back up again. So there's a small interstitial deletion here. Now, this is whole genome sequencing data, so you can also look for rearrangements. And it turns out that there's a rearrangement that links this left side of the deletion to the right side of the deletion, right? And if you think about it, that makes a lot of sense, that it almost had to be, because if you have a deletion of this, that leaves, this end of the DNA and this end of the DNA sort of hanging out in space. And the chromosome sort of won't allow it. It'll start getting munched away. And so each of these things needs to attach to something. In this case, they attached to each other. And so you could see the rearrangement between these 2 loci. It could have equally well happened that they would have attached to some other locus, each of them, and you could have had a more complex sort of event. But regardless, this sort of indicates that this deletion had to have been accompanied by a rearrangement that at least one rearrangement that involved its breakpoints. And so its boundaries were being determined by that the rearrangements that caused it, right? And this has to be true for every interstitial deletion and every interstitial amplification. Every event that covers actually less than a chromosome, at least less than a chromosome, right? That you end up having an end to the DNA, an unnatural end to the DNA that needs to be dealt with. So what determines the boundaries of rearrangements? Well, this is a view of the rearrangement density across about 3,000 cancers from PCOG, where we're going from chromosome 1 along each axis, 1 to chromosome X along each axis. So it's a 2-dimensional view. And the idea here is that, say, a dot in this top box here indicates a rearrangement between a locus on chromosome 1 with a locus on chromosome X. And so on with the other chromosomes. So the thing that jumps out is that, well, where do you have the connections between different, genomic loci most frequently? What jumps out is the shortest events are the most frequent, right? If you look at events that are near the X equals Y axis, from chromosome 1 to chromosome 1 or chromosome X to chromosome X and so on, those are occurring much more frequently than events anywhere between chromosomes, or even between distant loci within a chromosome. So why is that? Well, this like mimics high C data very closely. In fact, 2,600 times more closely than anything else, high C data predict this. So this is a view on the right of high C data from a cell line. And again, you see that the most frequent contacts in the high C data are within chromosomes. And so, what's the point of high C data? Well, it's to determine the 3D conformation of the DNA in the cell's nucleus, right? And it's giving a result that really mimics rearrangements. Well, if you think about it, it makes a lot of sense, right? How do you generate high C data? Well, people generate it by fixing the DNA in a cell, and that causes cross-links between the DNA, and then they fragment that DNA so that they can collect the cross-linked DNA and sequence across the cross-links. And so then they say, well, whatever got cross-linked together is close to it, our neighbors. So what they're doing is they're generating rearrangements in the cell by with formalin, and then sequencing across them. In the left, the cancers are generating the rearrangements, and we're sequencing across them. So it makes sense that that cancer rearrangements should follow the three-dimensional conformation of DNA. It's essentially because it's like cancers are doing a high C experiment, right? So that allows us to predict the boundaries of rearrangements, and therefore the boundaries of copy number alterations. And the result of that is the length distribution of copy number alterations. This is something that we identified, many years ago, that the most frequent copy number alterations are the shortest ones, and they decrease in frequency as the length increases. Approximately 1 over length, within a relationship of approximately 1 over the length of the event. And the exceptions are events that cover a whole chromosome arm or 2 whole chromosome arms, where there are different mechanisms that come in. Centromeres are easily breakable, causing whole chromosome arm events, and whole chromosome events can be caused by sort of non-destruction events. They don't need rearrangements. But this sort of 1 over L relationship very closely mimics the relationship between, the three-dimensional distance between loci as you go, as you make them more distant in the genome. Now, this 1 over L relationship, relates really to interstitial events, the kinds that I was talking about in the earlier slide with that little deletion, where both boundaries are within the chromosome arm. And so that's shown by this green line here. There are also events that start at the telomere or centromere and end, within the chromosome arm. Those don't need to follow the same high C kind of relationship because they don't need to connect to things in quite the same way, right? If you have a telomere bounded event, what can happen is you just have a cut in that chromosome, and a piece floats off, and then maybe it gets capped. And so it doesn't need to connect to an adjacent site so cleanly. And so the result is that events that start at the telomere or start at the centromere can end anywhere within the chromosome arm with approximately equal likelihood. So that's sort of more uniform distribution with a little bit of an uptick at the ends. So when trying to understand, how events are bounded, it turns out that it's easier to deal with telomere bounded and centromere bounded events because you're dealing with a uniform distribution rather than the sort of more complicated 1 over length distribution. And so we took that into account and developed Biscuit, which we published on a few years ago. So I'll just describe it briefly. And the idea is that, well, we know that in the absence of selection, a telomeric event should end anywhere in the chromosome arm with approximately equal likelihood. And so here's the sort of distribution of boundary locations in the chromosome arm that you'd expect. And this is the cumulative distribution, where none of the events ended by chromosome by the telomere. And then as you go along the chromosome arm, more and more of the events ended by that point, until all of the events that started the telomere ended by the centromere. So this is the cumulative distribution. And we see this kind of pattern in some chromosome arms. So an example is 20p, where it's mostly uniform. There's a little bit of an uptick here, but you remember in the prior slide that there was an uptick in the background distribution as well. So it's pretty close to the background distribution. So we see no evidence of selection going on here. In contrast, if you looking at a boundary analysis of in this type of chromosome arm, where you have very few events that end within the early parts of the chromosome arm, but then there's this jump in the frequency of breakpoints at this locus, right? That's indicating that the cancer likes deleting whatever it is at this locus here, right? So events that include that locus get selected for, and so you have this jump, and events that exclude it get selected against. And the cumulative distribution looks like this. So we see that in 9p, for instance, where we see very few short deletions, but then when you hit this locus here, all of a sudden you have a jump in frequency, and that jump is exactly where p16 lies. So it's identifying, positive selection for deletions of p16. In contrast, there can be chromosome arms where you have a lot of short events, but as soon as you pass this locus, the number of events drops, indicating that the cancers don't want to have events that include this locus, right? So that's an indication of negative selection. And an example is here, where you see actually quite a lot of deletions of 8q, short deletions of 8q, but as soon as you pass this locus, you see very few deletions of 8q. And these are hemizygous deletions, so one copy is left, but you see very few of them. And this locus is where MYC lies. So we're seeing actually negative selection against loss of MYC, which is a different thing from showing that's a driver gene. It's showing that's also sort of an essential gene in many cancers.
Hi, Dr. Baroukhim.
Yeah.
I have a question about this uniform background of the breakpoint. So is that related to certain mechanism, how the break can happen?
Yeah, so I think it really is that, like, you have you can have a break anywhere in the chromosome arm, and then and the rest of it floats off, and then you just have that bit capped, and the rest of the chromosome survives. So you don't need to connect to something like nearby. Whereas you have a break in the chromosome arm, and that bit floats off and connects to another region of the genome somewhere else, and then gets pulled along. But it's it's not, it doesn't require the same sort of events as the interstitial amplifications or deletions, where you need to sort of connect to each other most frequently.
I was thinking like breakage fusion bridge cycles and some other I was thinking if there's any physical like tendency that, because if I think about it, like the thing close, let's say start from the telomere, I was thinking like the breakpoint that close to telomere might be easier to
so that's a great point. So a breakage fusion bridge cycle, you have the fusion between two, well, I don't know, a break somewhere and then a fusion, right? And that break could have occurred anywhere within the chromosome arm, and then you get the fusion. And then after that, you can get further successive breaks as you're pulling the chromosomes apart. And that again could happen anywhere within the chromosome arm. And so what actually can happen is that you can you could start with a lot of the chromosome arm, and then you'd start munching down and like lose a lot of the chromosome arm on one side. or gaining, that bit of increasing amplification of that chromosome arm on the other side. So that this cycling of events that keep sort of munching off bits of a chromosome arm may account for the sort of smile appearance that changes it from the uniform distribution. So that first event, I think can tend to occur if you connect sort of two telomeres, right? And then you pull them apart and it breaks. It can break anywhere between the two centromeres. And so that's sort of more uniform. But then you sort of add in these second order events and so on. Did that
Yeah,
address?
that's very, very clear. Thanks.
It's a stick breaking analogy. And one of our reviewers is the one who came up with it. Great. So we use this method to identify regions of positive and negative selection across TCGA 11,000 cancer samples. And here we show on the left loci that we thought were subject to positive selection, to either deletions on the left or amplifications on the right. And then on the right, we see effects of negative selection against deletions on the left and amplifications on the right again. So here we have on the far left is tumor suppressor-like loci. Then next to that is oncogene-like loci. And then it's essential gene-like loci. And then it's toxic gene-like loci, right? So when we looked at those loci, we found that they overlapped with known cosmic genes quite well, significantly, but we identified a lot of unknown genes, and I'm not going to go into details about that. We also, actually found more loci under negative selection than positive selection. And when you look at deletions, using that same DNDS ratio that Martin Carena and Peter Campbell came up with before, we find that loci that we think are under positive selection have positive DNDS ratios, like you would expect, and loci under negative selection have negative DNDS ratios, like you'd expect, at least for the nonsense splice site and truncating mutations, which indicates that we really are identifying loci that are negatively selected, actually, even for mutations. And moreover, when we could assign a fitness value to each of the loci, so that if you in this case amplify the locus, how much negative fitness does that cause to you? And looking at across chromosome arms, the ones that have the greatest negative fitness scores have the lowest frequency of amplification, and vice versa. And the same is true for deletions. And so we do see that negative selection is heavily shaping cancer genomes. So at least copy number alterations in cancer genomes. But this is I like this, but it's we didn't want to end there, because we're missing out on all those interstitial events. And so the focal copy number alterations, which in some ways are the most illuminating, because they can span many different amplitudes, from homozygous deletions to high-level amplifications, and they have two ends, and each of those ends, boundaries, is useful. So we've now developed a method called Trisket that addresses these. And the method it uses is it determines the possible histories that generated each copy number profile, calculates how unlikely these histories were, and then uses these surprise factors to detect evidence of evolutionary selection. And before I move on, I just should say that when anyone who knows me knows that I'm actually quite incompetent. So anytime I say that something that we've done, it's somebody else who's done it. And in this case, it's Shahab Sarmashki, a postdoc in my lab, and Ellie Kim, very, very talented undergrad who's now a graduate student here. And so I'll describe what they did. I won't describe how we determine the possible histories that generated each copy number profile. It's too technical, but I'll describe the sort of fundamental thought process. And so the idea is that, well, let's say we identify a set of events that occurred in a cancer sample, right? So in this case, it was 3 events on a given on this chromosome arm. And the result was at locus X had a 1 copy amplification. Here we're looking at mom's chromosome only, so it should have had 1 copy, and in this case it had 2. Right, so we were at 2 copies. Well, how surprising was that? Well, we know what the length distribution of each of these events should be, right? We know that this is a telomeric event. It should have ended anywhere in the chromosome arm with approximately equal probability. This is an interstitial event, so it follows that 1 over L distribution. And this is a centromeric event. Again, it's a uniform, faced by uniform distribution. So if you look at event 1, right, we would expect that, well, 40% of the time it should have gone past X. X is 60% along the chromosome arm. And 60% of the time it would have ended beforehand. So it had a 60% chance of not covering X. We do the same thing for each of these events, and we find that there was a 60% chance that this event covered X, a 10% chance that this event did, and a 40% chance that that one did. So, or should have, right? So you could then say, well, under alternative histories, what is the likelihood of different amplitudes of amplification, right? So, for instance, you could have had 40% of the time you would have had this event cover X, and then maybe 90% of the time you would not have had this event cover X, and 60% of the time you would have had this one that covered X. So you would have had 3 copies, 40% times 90% times 60% of the time, right? And which is like about 20%. 21.6%. And so you can do that for every possible combination, right? And you can say that, well, you would have expected that cancers that had 3 events of that in that fashion should have had 5 copies 2% of the time, 4 copies actually 1.6 plus 3.6% of the time, and so on. We observed 2 copies, right? And so that would have been expected about 40% of the time. Great. Now, but if you do this in 1,000 cancers and you get these sorts of numbers for each of those 1,000 cancers, and you observe that, well, actually, level 5 was achieved 10% of the time, not 2.4% of the time. That tells you that there was positive selection going on for level 5 in particular. Whereas, maybe level 3, you expected that it should have occurred almost 50% of the time, and you only observed it 20% of the time. Well, that's negative selection against level 3, right? So we can look at actually selection, positive or negative selection for every amplitude level at every locus in the genome, taking into account every copy number alteration in that sample. So we've run that now on the same 11,000 TCGA samples, and we're trying to extend to larger data sets. At the moment, actually, we have more. So at the moment, we've detected 151 positive selection peaks and 106 negative selection peaks. And that extends from homozygous deletions to hemizygous loss to these, copy neutral events, and then gains in high-level amplifications. It's interesting that actually we're seeing a lot of positive selection for hemizygous loss, for example, which it's been very difficult to detect in the past, right? Like identifying regions that want to be hemizygously deleted rather than homozygously deleted. So how do we do this? Well, this is an example of our output where we're looking at homozygous deletions versus 1 copy, 2 copies, 3 copies, 4 and 5, right? And for each of these, we have the location on the genome on the vertical axis from the telomere at the bottom to the centromere at the top, with the p-values, the significance along the horizontal axis, going with p-values favoring positive selection to the right and negative selection to the left. So this is looking at 9p. And what we find is that there's strong positive selection for homozygous loss at this locus, which centers on p15, right? Which makes sense. p15 is a tumor suppressor gene. Great. then we take that into account and say, okay, now we know that P15 wants to be deleted. So understanding that we will expect to have a high rate of P15 deletions, what do we see next? What's the next most surprising thing? Well, then the surprising thing is RPS6 is almost never deleted, right? RPS6 is actually a ribosomal protein. So it's there's the algorithm is saying there's strong negative selection against homozygous deletions of RPS6. And it makes sense, actually. RPS6 really is an essential gene. So we are, I think, identifying true negative selection there. Okay, great. So recognizing that RPS6 and P15, what's the next most surprising thing? Well, the next most surprising thing is that the P15 deletions were longer on average than we'd expected in the northward direction, right? And so they extend past P15 more often than you'd expect, P16 more often than you'd expect, right? So we're seeing positive selection separately for P15 and P16. So disentangling this locus. And then recognizing that P16 wants to be deleted, what's the next thing that we detect? Well, it's that homozygous deletions of SMU1 are negatively selected. Cancers don't want to delete SMU1, right? So we're really systematically disentangling the positive and negative selection at this locus. Now, this was all at the homozygous deletion level, right? Where P16, P15 we already knew about, but we also are identifying strong signals at other levels. So, for example, when you do the analysis in just squamous cell cancers across all squamous cell cancers, we find actually no positive selection for homozygous deletions on 3P, but strong positive selection for hemizygous losses on 3P at this locus that encompasses PDZ-RN3 and three other genes. It turns out that PDZ-RN3 is an E3 ubiquitin ligase that is actually a degradation target of HPV, human papillomavirus. And HPV degrades PDZ-RN3. It's a degradation target. And so there's, so it's possible that PDZ-RN3 is an actual haploinsufficient tumor suppressor gene here. Interestingly, HPV-induced squamous cell cancers tend not to have 3P loss. 3P loss occurs in 75% of squamous cell cancers, but not the ones with HPV loss, presumably because they're suppressing PDZ-RN3 through an alternative mechanism, right? So we don't know that PDZ-RN3 is a haploinsufficient tumor suppressor gene. This is sort of a recent finding, but we now have collaborators working on it, and we found many dozens of other events like this that we have yet to disentangle. So that's it for this. I have a few more slides, but I won't describe them in detail, except to say that looking at rearrangements in the same sort of analysis of the two-dimensional analysis of where rearrangements start and end, as well as the one-dimensional analysis of where the breakpoints are, we identified a locus that's frequently rearranged with deletions in a gene called BRD4. BRD4 is a known actually oncogene and therapeutic target, and yet we're seeing deletions that actually in the gene that decrease its expression levels. These deletions tend to occur in the context of gains of the whole chromosome arm. So what's going on is that you're having a gain of the whole chromosome arm that seems to be targeting cyclin E1, and then when that happens, you get a deletion of one of those copies of BRD4 in particular. And so that all comes down, suggests a Goldilocks situation where you've got where the whole chromosome amplification leads to increased expression of BRD4, which is toxic, and the deletion leads to reduction of that toxicity. And these folks led this study and actually showed that overexpressing BRD4 leads to decreased rates of cell growth. Deleting BRD4 also leads to decreased rates of cell growth. But when you delete BRD4 in the context, delete one copy of BRD4 in the context of overexpression when you overexpress and then delete the endogenous BRD4, you rescue from the suppression of overexpression. And so that this is the first example of like a focused rearrangement targeting overexpression of toxic gene. So anyway, I'm going to end there with the overarching concepts being that negative selection is infrequent for point mutations as far as we know, although I think we may have more to discover there. But common for copy number alterations. And it's useful both to understand positive selection, and I didn't get into this, but it has the potential for indicating therapeutic targets. And to detect negative selection, you have to understand, copy number alteration lengths and boundaries, what causes them, and what they imply for what's being selected in the cancer genome. And I just want to thank the folks in my lab, current folks, as well as alumni, as well as many collaborating labs. Thank you for listening, and open up to any additional questions.
Thank you, Dr. Baroukhim. It's wonderful. Any questions?
I'll start with a couple more if you don't mind, while other people hopefully think. Thanks for a great talk. I appreciate it. I might want to ask a focus question, and sorry to make it again kind of clinically focused for molecular pathology, but then I have a broader question. When you've looked at, excuse me, I've kind of lost my voice, at the 9p locus and all the great work you did to disentangle that, one of the emerging biomarkers on that locus now is MTAP. And there's a lot going on in terms of disentangling the breadth of deletions in that locus that involve CDKN2A2B, that involve MTAP. Our colleagues in AP use MTAP as a surrogate marker for CDKN2A deletion at an IHC level, since P16 is a real hard target at a physiologic level. What else have you learned there?
Yeah, it's a great question. So the idea there is that loss of MTAP leads to a synthetic lethality with loss of PRMT5, right?
Yeah.
So here we're asking, is there negative selection against MTAP alone? We haven't detected that, right? The boundaries are not focusing on MTAP, and MTAP is often homozygously deleted. But the real analysis we want to be doing is among those cancers that have loss of MTAP, do they have negative selection against loss of PRMT5? And do they have more negative selection against loss of PRMT5 than cancers without loss of MTAP, right? So
yeah.
It's a slightly different question. It's a different formulation. And so we're working on methods to really address that in a formally, statistically rigorous way. And the hope there is that then we can use this sort of approach to detect synthetic lethalities.
That would be really cool.
The cool thing about that, I think, would be that current methods to detect synthetic lethalities are essentially you do a CRISPR screen in a cell line or in a mouse model, and you look for signals in cell death or cell growth rates and things. But here we have the opportunity to look for synthetic lethalities where the signal is human tumor formation in the human context, not in the cell line context or the mouse context, and in the context of not just growth assays, but in the presence of the immune system, vasculature, and so on. So anything that would alter the evolution of that tumor would be impacting it. And so it has the potential to identify things that we're not really focusing on in the lab.
Fascinating. I'll let others ask questions. I'll hold my other one.
Great.
Yeah. This is Betsy. Can you hear me?
Yeah.
Hi. So I have a question regarding copy neutral loss of heterozygosity. So frequently when we see the biallelic loss of P16 and on 9p, contiguous to that region is a copy number neutral loss of heterozygosity that extends much further into the P arm. And, you know, we normally think about copy neutral loss of heterozygosity if it occurs, let's say, by itself without an adjacent deletion, as potentially harboring a gene that is mutated, and then you get a biallelic mutation of that. But I'm wondering, given your thoughts about negative selection, if really the copy neutral loss of heterozygosity is a way of actually rescuing that deletion of those essential genes that cannot occur. Just your thoughts on that.
Yeah, absolutely. So, yes, the second. So, across all cancers, chromosome 17p is, I think, the most frequently lost chromosome arm. P53 resides on 17p, right? And it's, it undergoes loss of heterozygosity with copy loss, right? In gliomas, 40% of gliomas have mutations in P53. Almost all of those have loss of heterozygosity. And almost all of the time, that's copy neutral loss of heterozygosity. Gliomas clearly don't like losing even one copy of 17p. Conversely, they have like 80% of them, at least glioblastomas, have lost chromosome 10q, right? Which includes P10, but only half of the time when they lose 10q do they lose the other copy of P10. So I don't think that P10 is actually necessarily the target. So they want to lose chromosome 10, and they don't want to lose they want to lose at least want to lose one copy of chromosome 10. They don't want to lose even one copy of 17p. Endometrial cancers have occasionally have P53 loss, and that, and they have loss of 17p with copy loss, but they frequently have P10 inactivation, and that's almost always due to copy neutral loss of heterozygosity of chromosome 10, right? So endometrial cancers don't want to lose even one copy of chromosome 10. So clearly, there's negative selection going on against copy loss, and it's tissue-specific negative selection. And that's something we don't understand, and I think is really cool. Did that address your question? You're muted if you can answer.
Betsy, you're still muted.
Sorry. Yeah.
Oh, great. Okay. Any other questions?
Thank you, Dr. Baroukhim. I have another question regarding this intricate relation between negative positive selection. And we know the copy number change can be caused by chromosomal instability. So it's an ongoing process in the cancer cell. So would this combination between negative positive selection suggest a more complicated trajectory, or let's say how the cancer cell can acquire these changes rather than just gain one or lose one and stop?
Yeah, I mean, I think that there's like ongoing selection where the tumor is likely to be tuning itself, right? With gains and losses that control both its positive and negative selection. And the BFBs you described as I think going to be an excellent example of that, where there's been a lot of work done on extrachromosomal DNA, where it's, I think you're essentially sort of generating a plasmid that has the most effective positive selection possible by getting rid of any negatively selected toxic genes, and then also including the exact regulatory elements at once and so on. And I think complex amplifications are likely to be doing that in general, even when they're not in extrachromosomal DNA. But we haven't really addressed that sort of ongoing evolution as well as we should. Did that address your question?
Yeah. I was very fascinated about this concept of negative selection, Actually, I was thinking, like, what causes that? We know that there's some dosage balance that is required by the cell, but I'm not sure what actually make them deleterious. Any idea?
Right. So we published another paper last year on toxic genes, and identified one toxic gene, for example, that is an RNA processing gene, and so we thought like it's involved in RNA splicing, and so the expectation was that by amplifying it, you'd lead to changes in RNA splicing. And we were surprised to find that that actually was not the case, that it had like additional effects on DNA damage repair, right? Where it was by amplifying it, you sort of push DNA damage repair towards, and actually, Amy, if you could mute, please. If you're pushing DNA damage repair towards NHEJ and away from homology-directed repair, or HR, and so causing more misrepairs, right? And so I think often it's this changing just the balance of important pathways in the cell that leads to the negative. So the cell is highly tuned, and so when you change the balance, you can cause growth disadvantages. And it was interesting actually as well was that it was not a massive change in the growth, right? When you overexpress the gene in cells, it harmed them, but it didn't really destroy them. It causes a medium level decrement in growth. So I think often these negative selection things, they're causing sort of slow effect, like small effects over long time periods, and so you're seeing that as well. Did that address your question?
Yeah, thanks a lot.
Yeah.
Wonderful.
Okay, we are over time. We're going to let Dr. Baroukhim leave for the day. Thank you, sir, for joining us today. Appreciate your time.
Thank you so much for inviting me. It's been wonderful speaking with you all.